All right, in this video, we are going to talk about the replication of DNA. And this is the third and final of the processes of the central dogma of molecular biology. Gene expression, which includes transcription and translation, and then the replication of DNA. So all three of these biochemical pathways involve the production of polymers with information. DNA has information, RNA has information, proteins have information. And so the production of these polymers requires a source of information. Where is that information coming from when the enzyme is putting together this macromolecule? So what is the template? So I have a series of questions that hopefully you have been asking yourself with regards to each one of these pathways that we've learned so far, transcription and translation. And now that we're at DNA replication, you're going to ask these same questions again. Okay, but now with regards to producing or making a new double-stranded molecule of DNA. So the basics of DNA replication in all organisms is that it is semiconservative. What that means is that we start off with a parental molecule of double-stranded DNA, and the two new molecules that we produce consist of both a parental strand, so an old parental strand, and a new daughter strand. An old parental strand and a new daughter strand. Okay, that's referred to as being semiconservative. The production of DNA replication here is bidirectional. So we start at a place called an origin of replication, and the two strands of DNA are replicated in both directions. And so we have what are called two replication forks that make up a replication bubble. And finally, like any other polynucleotide, synthesis progresses in a 5' to 3' direction. No matter whether we're talking about RNA or a strand of DNA, synthesis always occurs in the 5' to 3' direction. The 5' to 3' direction. Now, DNA polymerase has certain requirements, and some of them are different than RNA polymerase. So DNA polymerase requires the building blocks here that are deoxyribonucleotides, okay, instead of simply ribonucleotides that RNA polymerase requires. So that would be an A, a G, a C, and a T, not a U. It requires a source of information, right? What are going to be the sequences of the nucleotides in this new strand that's being made? Where does that information come from? It comes from the template strand, so one of the two parental strands of the original DNA molecule. And finally, and what's unique to DNA polymerase is that it requires a primer. DNA polymerases cannot just take two nucleotides, slap them together, and go from that. They need an already established primer, so maybe 10 to 20 bases. And we'll talk about how those primers are made in a moment. And so what that DNA polymerase will do is it recognizes this sort of double-stranded, single-stranded junction made right here near the primer, binds, and then starts reading and writing and reading and writing. DNA polymerase is dependent on magnesium ions. So these are going to be your metallic cofactors here. So we have a magnesium ion here, we have a magnesium ion here. Those magnesium ions are brought into the active site by some aspartates here. And the purpose of these magnesium ions are to properly orient the incoming nucleotide. So these positive charges interact with the negative charges on that incoming DNTP, such that the proper phosphate that's going to be attacked here by the 3' hydroxyl group of the end of that growing strand is in just the right place. DNA polymerase is also dependent on another protein called a sliding clamp. Now, this sliding clamp, as you can see, is topologically linked to the molecule of DNA and interacts with the DNA polymerase. And what this does is it prevents the DNA polymerase from falling off the template. Okay, what it does is it increases the processivity. Now, processivity means that what we have here is a situation where our DNA polymerase doesn't just bind to the template, add a nucleotide, and then fall off. And then bind to the template, add another nucleotide, and then fall off. Instead, it stays bound to the template. It stays bound to the substrate, and it keeps adding nucleotides over and over and over again. And it's in part able to be this processive because it has this, it's interacting with this sliding clamp that keeps it from falling off. And so with the sliding clamp, it can process over 10,000 base pairs before falling off. Without the sliding clamp, it falls off within 20 to 100 base pairs. Now, E. coli is part, the DNA polymerase 3 within prokaryotes, specifically E. coli, exists in a fairly large complex where we have actually 2 DNA polymerases here, one that's going to be synthesizing the leading strand, one that's going to be synthesizing the lagging strand. And they are connected to a common clamp loader. And this clamp loader is what is going to be responsible for topologically linking these sliding clamps around the DNA. And those, of course, then are going to interact with the DNA polymerase and keep them processive. So we've got this large complex that you're going to see that exists at each of the DNA replication forks. Now, DNA replication occurs at what are called the origins of replication. And if we are talking about a eukaryotic chromosome, there are going to be many origins of replication within each individual chromosome. Okay, if that wasn't the case, DNA replication would take far too long. A origin of replication in E. coli looks like this. Now, you don't have to memorize all of these various sequences, but there are 2 that are important. First, these sequences here that are going to be bound by a protein called DNA. And then this sequence here called the DNA unwinding element, or the DUE, or just the DO. Notice there's lots of T's in this sequence, and that's on purpose because this is going to be the original sequence that is going to unwind to separate the 2 strands of DNA so DNA polymerase can get in there and start reading those 2 strands separately. So what we have is that DNA A is going to bind to these sequences here shown in gray. They are going to be what's responsible for separating the 2 strands of DNA at the DO region. And DNA B is going to be recruited by DNA C to these sequences, and they are helicases. So this DNA B is a helicase that's responsible for, in this case, this replication. So this is going to be our beginning replication bubble consisting of 2 replication forks. This one right here and this one right here. And so you have one DNA B at each replication fork, one helicase at each replication fork, and their job is to keep separating those 2 strands of DNA, separating those 2 strands of DNA. Now, as replication occurs, there are lots of different proteins that are going to be necessary. We just got finished talking about DNA A, B, and C, but we're going to get into a primase. That's the enzyme necessary for building an RNA primer. We're going to have single-stranded binding proteins. Those are proteins that bind to single-stranded DNA and stops the DNA from coming back together again before it gets replicated. And finally, we have a gyrase. We have a topoisomerase that's necessary for dealing with the supercoiling that's happening as a result of DNA replication and the separation of the two strands of DNA. So there's a lot of proteins involved in DNA replication. Now here is a replication fork. So we would have one of those DNA polymerase complexes at this fork here that consists of one DNA polymerase that's transcribing this leading strand here, or not transcribing, excuse me, replicating this leading strand, and one that is synthesizing this lagging strand. How do we know which one's the leading strand and which one's the lagging strand? The way you figure that out is you look at each strand that's being made, okay, so both in red here. The strand that is being made in the 5' to 3' direction into and in the same direction that that replication fork is moving, that's the leading strand. The strand that's being replicated in the opposite direction that this replication fork is moving is called the lagging strand. So make sure that you can differentiate the two. The replication of the leading strand is fairly straightforward, okay? So at the origin of replication, when the two strands are separated, the next step is that DNA primase is going to lay down a primer, okay? And it's an RNA primer because RNA polymerases don't require primers themselves. So this is the way we can get a primer down. But what that does mean is eventually that RNA primer is going to have to be pulled out and replaced with DNA. Okay, so regardless of whether we are talking about the leading strand or the lagging strand, we need to lay down a primer. Now for the leading strands, the strands that are being made in the same direction that the replication fork is going, so the leading strand here, and then the leading strand going in this direction here, no problem. The DNA polymerase recognizes the primer, binds to it, reads, writes, reads, writes, reads, writes, reads, writes. RNA primer binds, DNA polymerase binds to it, reads, writes, reads, writes, reads, writes, reads, writes. Now, that DNA polymerase is just going to keep going and going and going until it reaches the end of the chromosome or it reaches another DNA polymerase coming in the opposite direction. So, leading strand, very straightforward. Lagging strand is a bit tricky. It occurs via this discontinuous replication. Okay, so like I said, even the lagging strand is going to start with the synthesis of an RNA primer. But notice, here's our replication fork. It's opening in the right to left direction, but that primer and everything after that primer is going to be being built in the the right to left direction. This is going to be from the left to right direction, and the opposite direction of the replication fork. So 5' to 3'. So in red, we have an RNA primer. Okay, that gets built. And then we have our DNA polymerase 3, right? This is the one that's important for most of the DNA synthesis. Recognizes the primer and builds, reads, writes, reads, writes, reads, writes. Notice again, it's going in the opposite direction of the replication fork. Where does this end? Well, it stops at the beginning of the previously laid down fragment. These are called Okazaki fragments. So here's our previously laid down Okazaki fragments. This is the one that we were working on, and it reads, writes, reads, writes, reads, writes, reads, writes until it bumps into the beginning of that previous Okazaki fragment. Done. Falls off. Okay? And then what we're going to have is we're going to have another primer that's going to be laid right up here. Primase is going to come here. It's going to lay down a primer, and then we have another, right? Read, write, read, write, read, write, read, write, read, write, until it hits the beginning of the next Okazaki fragment. So we're going to have hundreds of these Okazaki fragments just constantly being made at this lagging strand. And now we have a problem. We need to do two things. Number one, we need to remove that RNA polymerase and replace it, or this RNA primer, and replace it with DNA. And then number two, we need to seal up the nicks between the adjacent Okazaki fragments. So let's see how that's done. Well, the job of removing and replacing the primer is done by another type of DNA polymerase called DNA polymerase 1. And like I said, it removes the RNA primer and adds DNA, but here's the problem. It's a DNA polymerase. Like all DNA polymerases, it needs a primer. It needs some end to start adding on to. Well, what is this going to use? It can't use another RNA primer because that defeats the whole purpose. So let's take a look at what that is. Well, what that is right here. When it removes this red, chew, 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 it uses right here. It's going to use the end. Okay, this 5' actually, this 5', it's a bad picture, is talking about the beginning of this primer. Right here at the end of the blue is actually 3', and it's going to, that's what it uses as a primer. The 3' end of the upstream Okazaki fragment. It just adds to that. But we have another problem. So it's reading, writing, reading, writing, reading, writing. When it gets rid of the last, it can't connect this Okazaki fragment to this Okazaki fragment. And that's the job of DNA ligase. So DNA ligase is an enzyme that seals the nick between two fragments of DNA. It adds that last phosphodiester bond. This is the same DNA ligase that we learned almost the first day of class, right? Necessary for recombinant DNA technology. We can cut DNA fragments with restriction endonucleases and then glue them back together with DNA ligases. And this is where we get them. Now, the elongation and the continued replication requires, as we sort of talked about earlier, several other proteins. Okay, we need that helicase. So here's that helicase, right here in green, that is responsible for separating the two strands of DNA. So that's going to continue on. It was laid down at the origin of replication, and it's going to continue moving and separating those two strands. We need these single-strand binding proteins to bind to the newly separated DNA strands, because otherwise they would just pop back together before they were replicated. And then this whole process here occurs in a very coordinated fashion. Right? The lagging strand, the name lagging makes it seem like the strand takes longer to replicate than the leading strand, but that's not the case. Because we have this nice coordinated process where we have everything connected. There's our DNA polymerase that's replicating the leading strand. Here's the one that's replicating the the lagging strand. We've got the primase coming in, laying down primers. Everything is coordinated. Everything interacts with everything else. And this is a very organized process. So take a look at this figure and make sure that you can name each one of the components that you see here. All right, in the next video, we're going to talk about the problem that is inherent with replicating the ends of linear chromosomes. And we'll talk a little bit about what that means and the ramifications of these problems.